Welcome to another Lipid Maps podcast. And I'm joined this morning by Andrea Lopez, who's the head of the Lipidomics facility at the Babraham Institute. Morning, Andrea.
Good morning, Matt. Thank you for inviting me.
And the Babraham Institute, of course, is on the outskirts of Cambridge and a few kilometres south of Cambridge, out in the very flat countryside.
It's absolutely very nice campus. We have 60 companies on site, biotech companies, spin-out companies. And the stress and the pressures of the job, there is a cycle path where you can skate as well. It's a mile long, but also you can go through the forest at the back of the institute, and there is the river Granta running along the lines of the back of the on the middle of the land that belongs to the institute. So it's a nice setting, and I think I love it for that reason.
Can you define lipidomics? It's one of the many omics we have in science.
Well, when I get to ask that question, and before I joined the Babraham Institute, I wasn't completely sure, what the omics and lipids will really mean. But when we started to use a tool called Bioplan, it made more sense to try to explain what lipidomics was. And lipidomics is, in essence, finding these connections and looking into what is changing into the lipid, not just the lipid composition, because we know the lipids have substrates and products into biosynthetic pathways, and they are interconnected. So those substrates and products are basically regulated by enzymes. And changes on those lipid levels might indicate some changes in the enzymes, which are then, being encoded, those enzymes, by genes. So something along the lines of the treatment or a drug that people has used in their experiments, or a knockout of certain genes, might alter this lipid biosynthesis. And explaining all those changes and the biological relevance of it, I think that's what defines lipidomics.
So the difference in lipids between, for instance, a healthy and a diseased patient, and linking it back to the proteome and the genome.
Absolutely.
For what that might indicate. And I guess, of course, a change in the lipidome could be indicative of a change in the enzyme content or the enzyme functionality, or it could be the change in the lipids themselves could be the cause of the disease. For instance.
We cannot tell if it's a change in the enzyme activity or in the enzyme amount. We just strongly suggest that enzyme is doing something related to the lipid pathway. It's very interesting because you were mentioning about a condition of and a treatment, but as the Babraham Institute is focusing on aging, we could also start talking about changes in the lipid biosynthesis in relating to age. So young individuals compared to age, different groups.
Yes, and that's something we don't tend to think of as disease, but there are definitely changes that affect all of us as we get older.
Absolutely, yes. Absolutely.
And we even see this currently with COVID, that old people are far more likely to be seriously affected than younger people.
And how does that lipid composition really is driving? Because most of the lipids are signaling molecules. So how this signaling is affecting the outcome, a positive outcome or a negative outcome. But it's not only the lipids by itself, it's all these components, as you were mentioning, the transcriptomics, the proteomics, and the lipidomics. And well, we also have metabolites in there. So as we know, lipids are also considered metabolites. So maybe you will correct me if I'm wrong, but 2/3 of the metabolites are lipids, or is 1/3 of the metabolites, Matt?
I don't know the number, but it's a very valid point. I think for people doing biology at school level, I know in the A-level syllabus here, so 16 to 18, lipids are purely structural molecules. They make cell membranes. And I think that's largely the extent to which it's covered. But as you said, they're vastly more complicated than that in terms of their roles in the cell.
Absolutely. And it's also very indicative of the state of for example, a cell. So whether we have a catabolic or an anabolic metabolism that happening at a certain time. Unfortunately, lipidomics is quite expensive technique to do because we're measuring at the facility, we're measuring at the moment 46 lipid subclasses, which composes around 800 molecular species. But we're talking about 800 molecular species in a replica. And with a single data point, you cannot really tell what is changing. You need statistics to be able to say what is significantly changing between the control condition and the treated one. So you need more than one replica. So the prices of it is costly. And each lipid subclasses have a different technique in terms of the chromatography to be able to separate them and to measure them with confidence. And it's not high throughput, it's comprehensive lipidomics that we do at the facility, which means quality is at the forefront of what we do, because we need to be absolutely sure that everything that we report in there is what it is.
If someone came to you with a sample or several samples and they were perfectly optimized for lipidomics, how long would it take to get the results? Is the analysis a day, a week, a year?
It's a very interesting question, Matt, because it does change depending on the sample. I mean, we do research here which haven't been done anywhere else. Some of the matrices, so let's call, for example, cerebrospinal fluid, they are scarce if they, for example, are even from a mice or from a human. So the amount of sample does matter for a lipidomics analysis. So if I have 10 microliters of plasma from a human individual, it's more easy to find more 10 microliters if you want to measure 46 lipid subclasses. But from a mice or from spinal fluid is slightly more difficult, the amount of sample. So if we talk about in terms of something that nobody has done before, we tend to do a test sample. It's essential that we know what we can measure, but also it's not a shot in the dark, but it's more targeted in terms of what we can do and read from the transcriptomics data. So most of the time, people come to us because they have seen their transcriptomics or their proteomics data pointed out to lipid metabolism. So we do an initial assessment of what is that the transcriptomics or proteomics is telling them. And we say, we better focus, instead of measuring everything at a huge cost, target the lipids that we're going to measure. But that doesn't mean...
So if you see, for instance, in the transcriptomics, differences in sterile metabolite genes, yes, then you would focus on the sterols rather than on fatty acids.
It is a very interesting point. So we have an internal collaborator here that we were seeing the transcriptomics pointed out to changes in the cholesterol biosynthesis. And when we measure the cholesterol levels, they weren't changing. So then you start to ask, what else is changing? What other metabolites or what other lipids are there to be able to measure, to see the whole picture? What is really happening, a consequence of that treatment? So indeed, we do some kind of targeted assessment, but by no means that we only focus on targeted analysis using mass spectrometry. So we have two different approaches, and our methods are, when we do the test samples, our methods are validated using high resolution mass accuracy, and also using a Q-trap instrument that will give you fragmentation. So we use both instruments to be able to nail down those changes in the lipid composition.
Once you've identified changes in lipid composition, what's the next step to analyze what's happening and linking it back to transcriptomics, for instance?
It's interesting that you mentioned that, because getting the results, the results from the machine, once the test sample has worked around really well, once we know how much sample you're going to give us, is it cells, how many cells they're going to give us, because the size of the cells also matters. It's not the same to do an analysis in a liver cell to an analysis in a T cell. So size of the cell does matter. The amount of tissue that we're going to use. Recently, we did some experiments in tumor tissue. And the interesting point about it is what part of the tissue from the tumor you're going to take, because we know tumor are necrotic at the center, but also you want to have like this biological relevance. What part of the tissue within it you're going to take? Are we going to homogenize the whole tissue? So it's a whole discussion and an initial assessment of how are we going to do this. So when we go and do the big experiments, as I call it, with the replicas, we know. So that usually takes, like, let's say 6 months to nail down that starting point. And it's essential that we do that for the confidence of the results. When we get into the big experiments and we have the results, the replicas, 800 molecular species per replica times 4 controls and 4 knockouts or treated or age condition, then we run them onto the mass spectrometry with chromatographic separation. And this will take around 2.5 to 3 weeks to obtain just these 46 lipid subclasses. The instruments run in parallel, as some of them have to run in serial. So it's absolutely crammed by the start of the project. Then we get the data, and we need to start calculating the relative levels of the lipids in those samples. And then it's a whole process before we get to calculate the relative levels, because we look at the data point by point. So if we obtain the data using our QTRAP system, so multiple reaction monitoring experiments, the process is not automated. We do it manually. We are confident that when we see a peak, it's a real peak, and it's not something that is being picked up by an algorithm. So the people that work at the facility have been trained for a long time to be able to distinguish what is a real signal and what is not.
And I think that's a very important point in many aspects of science. Don't throw your data into a computer and blindly believe the results that come out.
Yeah, and needless to say, we need quality controls in there. So we have a quality control sample that we know how it behaves, that we know the levels that we have been using since the years I took over to run the facility. So we know, what is expected from that sample. And if that sample doesn't behave really well, then we know something has gone on. And that's the first thing we do before running any sample is QCing the machine, QCing what is in there, running the stock solution from the internal standards. Is my retention time consistent? So quality control is key in what we do.
Absolutely. Could you summarize for us the steps of the lipidomics experimental process?
Right, Matt. Well, initially, when people come to us, they come with a research question that they might know what the proteome or transcriptomics is pointing out in terms of the genes that might be changing. And sometimes some of these genes are involved in lipid metabolism. So that's one step to try to approach how to do the lipidomics. And then we see from those transcriptomic results what kind of lipids we currently measure. It's also important the type and the quantity of the sample. If you want to look for a lot on lipids, the quantity of the sample is important because it needs to be above the limit of detection of the mass spectrometers. If you don't have any idea of what the transcriptomics or proteomics because you haven't done it, then we can start looking at the major phospholipids based also in the type of tissue, type of cell that the research question is based. We can then start to propose on certain types of lipid subclasses that could be follow-up. And that kind of give you a different approach of how to then build this relative quantitation of the lipids. And remember, in lipidomics, we always need a control sample and a conditioned sample that has changed, a knockout or a treated or aging, as we were discussing before. So when finally we got these relative quantitation results, we then start to build these interesting biosynthetic patterns to be able to use our free tool in Lipid Maps Biopan that built up all the lipid network for the lipid biosynthesis, correlating it with the genes that activate or suppress enzymes involved in lipid metabolism.
I think you've given us a really nice introduction to the whole field of lipidomics, which spans everything from wet biochemistry through to bioinformatics and statistics. So it really is the whole gamut of science.
Absolutely. But I think at the core is analytical chemistry, is mass spectrometry.
So Andrea Lopez, it's been a fascinating insight into lipidomics. Thank you very much, and join us next time for the next Lipid Maps podcast.